Hello and welcome back everyone to the biology class of Fun Scholar. So today I am Shushmita ma'am back with the second part of the lecture on the chapter the cell. So in the last lecture we had been discussing about the introduction to the cell. We have learned about the discovery, the scientists who actually discovered about the existence of the cell. We have learned about the microscope as well. So now in this lecture we are going to delve deep into the internal structure of the cell individually in detail. So therefore let's begin. So the first thing that we are going to deal today is about the structural components. So what do we understand by the term structural components? So if we see, or we already might know from our previous knowledge, that there are many cell organelles which are an important part and parcel of the cellular structure. But there are certain types of typical important we can say parts without which the cell cannot exist altogether. What are those parts? So therefore the parts which are essential for the existence of the cell, we can say, so though there are basically three of them. First one is the cell membrane. It is also known as the plasma membrane. Right. So if the membrane is not there, there is no boundary, there is no outer protective layer to protect the cell from any mechanical injury or from any invasion by the pathogens. So therefore, the existence of the cell will be jeopardized. Now, the second most vital structural component for the cell is the nucleus, which is actually called the brain of the cell. So this organelle is the one which gives direction to all the activities going on inside the cell. So even the formation of the new cell from the existing cell, that means the cell division, and also all the other functions carried out by the different cellular organelles, they are all coordinated by the instructions given by the nucleus. So therefore, if the brain is not there, so the sequence as well as the, we can say, the perfect working of all the parts of the cell will not be so much organized. And the last one of the structural component is the cytoplasm. What is cytoplasm? Cytoplasm is the jelly-like matrix of the cell. So it is like a semi-solid fluid in which all the cellular organelles remain immersed. It provides as the platform on which all the metabolic reactions of the cell take place. So therefore it is the medium for all the activities, just like air acts as the medium for the transferring of the sound from one place to another. Similarly, cytoplasm acts as the medium for conducting all the metabolic reactions of the cell. So now let's watch a video regarding the structural components of the cell.
In our last video, we studied the discovery of the cell. Wouldn't it be interesting to see how a cell looks? Let us do an experiment here. To do the experiment, we need a microscope, a slide, cover slip, iodine solution, water, forceps, and an onion peel. It's quite a few things. Trust me, it'll be worth it. Now take the onion peel and with the help of the forceps, pull away the thin lining from the inner surface. Now we are ready to begin. We first add a drop of water on the slide and then place the onion peel on it. Do not touch it with your fingers. Next, we add 2 drops of dilute iodine solution. Do you know why we add iodine solution to this specimen? We do this so that we can clearly see the parts of the cell. Else it is difficult to study them as they are transparent and it would also be difficult to differentiate between the parts of the cells. Now place a cover slip in this manner and make sure that no air bubbles are formed in the specimen. With this, our slide is ready. This entire process of setting up the microscope along with the slide is called as mounting. It is also important to see that there is enough light falling on the mirror of the microscope. Shall we view it now? Here we go. Now let us see our specimen. Wow, what do we have here? Cells with distinct borders separating each one from the other. Here somewhat at the center, there is a relatively darker stain. The rest of the space is lighter and has these tiny dark spots scattered all over. What are these spots and how do we understand them? We will see that in the next part of the video.
So now from this video, what have we come to know? Let's discuss a bit. We have come to know that if we do the experiment with the onion peel cell, so by staining with iodine solution or with safranin solution also, safranin dye, we can stain that. So we can see the basic structural components of the cell about which I have discussed before the video. That means we can clearly visualize the cell wall because onion cell is obviously a plant cell. So cell wall will be there which can be clearly observed under microscope and also the distinct peripheral nucleus. So these are the two main structures and obviously the cytoplasm will be there inside the cell since they are the living cells. So these three structural components will be visible under the microscope with this simple staining technique or I can say the mounting technique. That means preparation of the slide just from the raw materials and staining it and then observing under the microscope. So now let's proceed. So you can see here, so the cells, all the cells which have common in them, they are this genetic material, cytoplasm, the cell membrane. Now, again we have now need to watch another video regarding the structural components. Here we will discuss in detail. So first let's watch the video and then we'll summarize the points that have been discussed in the video.
In the previous part, we saw how an onion peel could be mounted, and this was the specimen we saw through the microscope. And in this part, we will cover the different parts of the cell. This outer wall here is called the cell wall. And notice that there is another layer on the inside of the cell wall. This is called the cell membrane. Yes, the outer wall is the cell wall, and the inner layer is called the cell membrane. Understanding these two is fun. Let's look at the cell membrane first. The cell membrane is also referred to as the plasma membrane. The plasma membrane is porous. Wait, what does porous mean? That means material can move in and out of the plasma membrane. Isn't that dangerous? Doesn't it mean that any harmful material can get in and affect the cell? That is a valid point, and hence there is one important detail here here we should be aware of. The plasma membrane is selectively permeable. Now, what does selectively permeable mean? It is like a guard standing at the office door. Only members with identity cards can go in and come out. In the same manner, the plasma membrane allows only specific material to go in and out of the cell. We will look at this in detail in our next video. What about cell walls? Do all cells have cell walls? Probably not. Did you know that an animal cell does not have one? An animal cell only has the plasma membrane and not the cell wall. Why is this so? To answer this, let me ask you a few questions. Where do plants grow? Do they have a house to shelter them against bad weather? Or can they approach the doctor for medicines against diseases? The answer to all these questions is a definite no. Plants have to protect themselves against adverse conditions like strong winds, temperature variations, and infections as well. Therefore, the plant cells have cell walls that act as a protector. This is a non-living rigid structure around the plasma membrane and is made up of cellulose. It not only acts as a protector but also gives shape to the plant cells. It also helps in cell-to-cell interaction and provides a barrier to undesirable macromolecules. It protects, it gives shape to the cells, and it helps in cell-to-cell interaction. Clearly, the cell wall is a very important part of the plant cell structure. Now look at this lighter region here. This lighter region here is the cytoplasm, which is a jelly-like substance in the cell space. Can you see these tiny parts in the cytoplasm? These are the organelles. What are they? Just like the human body has different organs, the cell has organelles. And just like the organs, each organelle has a different function. We will look at the organelles and their functions in another video.
So here again in this video, they have discussed about the structural components, but this time in details, that specifically what is the need of each type of structural component such as the cell wall, the cell membrane, and the cytoplasm. So therefore, they individually play an important role in the life of the organism, so or the cell. So now we will also try to understand that we have already learned from the video that cell wall is necessary for plant cell because they don't have a house, they don't have a medical facility to take care of them, such like as the other animals can do, because they can locomote from one place to another. So that is why they have cell wall for their extra protection. Now, apart from the structural components, other components are there which are known as cell organelles. Now, when organelles means they are the suborders of the organs. So they are below the level of organs. So that is why they are known as organelles. So just like that, there are many organelles such as the lysosome, mitochondria, plastids, ribosomes, about which we need to now know one by one in detail. So the first one is the cell membrane. So cell membrane you have already seen in the video also. The main functions of the cell membrane is to enclose the components, as I already told you. It is the outer covering and its function is protective in nature. And also it monitors what goes in and out of the cell. Why? Because it is selectively permeable in nature. It is selectively permeable in nature, and it allows certain particles out, for example the waste materials, out of the cells after the metabolism. Otherwise the toxic environment of the cell could not be prevented if there would have been accumulation of the toxic waste substances after the metabolism. Similarly, it also allows the nutrients to enter the cell and also the respiratory gases, so that the cell can also get the nutrition, can respire and continue the proper functioning. This layer of the cell membrane, mainly it is made up of proteins and lipids. It is known as the PLP structure, that means the protein lipid protein structure, right, in which the proteins are hydrophilic in nature, that means they can react with water, so they are present on either side of the cell membrane. On the other hand, the lipid which is sandwiched between the two protein layers, that is hydrophobic in nature, that is why it actually repels water, so it remains sandwiched in between. So this PLP structure is actually the main enlarged structure of the cell membrane. Now coming to cell wall. Cell wall, which is another cell organelle discussed in the video, that is made up of cellulose in case of plant cell, because for other types of organisms such as the fungus, the cell wall is made up of chitin, and for the chlamydomonas algae, the cell wall is made up of peptidoglycan. So the composition of the cell wall varies from species to species, but the nature and also the functions remain almost the same, that is protection from the harsh environmental conditions and also protection against the mechanical injury, then protection from the invasive pathogens. So you can see here that protective layer of the plant cell is the cell wall. It provides mechanical strength, support, and it is the reason why the actually the cells which have got a cell wall, they have got a definite shape. They have got a definite shape due to this property, that mechanical strength and support. Now, unlike the cell membrane, the cell wall is freely permeable, right? The cell wall has got, you can see here, they have also got passages within them. Those passages are known as plasmodesmata. Now, these passages help in the continuous flow of substances such as water with dissolved minerals, which is absorbed by the root cells from the soil, to move in a continuous column-like manner from the roots to upward. So because they flow from one cell to another by passing through this plasmodesmata from within the cell walls only. So that is why they are freely permeable, so that the important molecules can pass through them, and they are composed of cellulose, I already told you, and along with that, in some cases they also have got deposition of lignin or suberin to make them water resistant and hard, and also providing more mechanical strength. So when the plant cells actually mature, they are old enough, so then such type of deposition takes place in the cell wall along with that of the cellulose to make them more hard and rigid in structure. So here we can see that in case of plant cell, both cell membrane and cell wall is present, but in case of animal cell, only the cell membrane is present. So now we will see how the structure and the functions of various cell organelles within the cell can be compared to that of a chocolate factory, and how each component of the cell represents with the corresponding component of the chocolate factory. We'll have that comparison one by one. So first is the nucleus. I already told you it is one of the structural components of the brain. One of the structural components of the cell. It is also called the brain of the cell, and if brain is not present, no activity can be coordinated within the cell. So it is a vital cellular organelle. So let's first watch this video, then we'll discuss about the nucleus in detail.
We know that cells are the structural and functional units of organisms. How do they carry out different functions though? We studied in our previous videos that cells have organelles and each of them has a specific function that they carry out. These organelles work like departments. Each department has its own functions and an area assigned to them. All eukaryotic cells have a membrane around these organelles. Do you know what the use of this membrane is? The membrane acts as a boundary. It separates the inner contents of the organelles from the surrounding cytoplasm. It's interesting to study how each organelle works. In this lesson, we will study the nucleus, which is the major cell organelle. Why would I call it the major cell organelle? The nucleus contains the genetic material of the cell. When the cell decides to form new cells, do you know what exactly happens? In cellular reproduction, the nucleus plays a very important role. Inside the nucleus, can you see the threads-like entangled structures? They are the chromatin fibers enclosed within the nucleus. Chromatin fibers during cell division get organized and form rod-shaped structures. These are called as chromosomes. We've heard this term so often. Chromosomes are seen only during cell division. What exactly are these chromosomes then? What do friends and family say when they see a baby? She looks exactly like her mother, or she's so much like her father. Similarly, a new cell looks like the cell it came from. How is this possible? Chromosomes contain genetic material in the form of deoxyribonucleic acid molecules. They also contain some proteins. The DNA molecules have the information of how the new cells must be organized and structured, which is why the new cells are quite similar to the parent cells. Do you know what the function segment of a DNA is called? They are called genes. Genes make up DNA molecules, and DNA molecules together with proteins make up ribosomes. The nucleus is also covered by nuclear membrane. Can you see how many layers it is made up of? The nuclear membrane is made up of 2 layers. The membrane also has pores. What are these pores for? Do they have a specific purpose? Yes, they do. Without these pores, the material cannot move from the inside of the nucleus to the cytoplasm. Thus, the nucleus is a very complex organelle, and in its absence, a cell cannot undergo cellular reproduction. We will study a few more organelles in our next video.
So therefore, from the video, we have got the idea that the nucleus, which is the brain of the cell, can be compared to the recipe book of the chocolate factory, because recipe book is the main thing from which how the chocolate will be made and also what will be the ingredients of that chocolate, so that can be understood. So therefore, everything is present in the recipe book only. Similarly, how the cell will function, what type of proteins that will be produced, and how and when the cell will undergo division, everything is coordinated by the nucleus, because in the nucleus we have got the genetic material, that means the chromatin fibers which coil up during the cell division and they form the chromosomes, and the chromosomes contain the DNA and DNA contain the genes. So therefore, and those genes code for certain proteins, and these proteins then actually carry out the functions which they are assigned for. So therefore, according to the type of protein that will be synthesized in the nucleus, the functions of the cells will be carried out accordingly. So in that way, they coordinate the functions of the metabolic reactions of the cell as a whole. Similar to the recipe book, which actually dictates that what will be the recipe, what will be the ingredients, what will be the process of making the chocolate. Now coming to the ribosomes. Ribosomes can be compared to the chefs in the chocolate factory, because just like chefs which actually do the manufacturing process or the production process of the cake according to the recipe written in the recipe book. So therefore, ribosomes also produce proteins according to the genetic code which is present in the nucleus. So therefore, in the nucleus, the recipe is there, and ribosomes are the chef who actually produce the proteins according to that instruction. The ribosomes are also associated with endoplasmic reticulum, and so therefore there are two types of endoplasmic reticulum that we'll now see. So one is the rough endoplasmic reticulum. So endoplasmic reticulum is of two types. One is smooth, one is rough. Now the smooth endoplasmic reticulum, it produces lipids, and the rough endoplasmic reticulum which has got ribosomes, so it has got that rough granular structure. So that is why the name rough endoplasmic reticulum. It produces it produces the proteins, and both these proteins and lipids are then transported to form the cell membrane. So then these are very essential components of the cell membrane. They are formed from these two raw materials only. So that is why endoplasmic reticulum is an important cell organelle, and also it acts as the production floor. Why? Because just like the production floor of the chocolate factory on which all the activities are going on, similarly the production floor of the endoplasmic reticulum helps to produce these two vital components of the cell membrane, that is the lipids and the proteins. Now we will understand about the endoplasmic reticulum with the help of this video.
In our last video, you must have studied the nucleus as an organelle. Now we look at an interesting duo of organelles. They are the endoplasmic reticulum and Golgi apparatus. Let us first look at how a factory works. A factory manufactures many things. Once the products are manufactured, they need to be assembled, modified, packed, and then transported. A similar process takes place in the cell. The endoplasmic reticulum works as a factory, whereas looking at the endoplasmic reticulum in detail. How does the endoplasmic reticulum look? Just imagine a long tube which is bound by a membrane. This tube is then placed in oblong bag-like structures which are placed around the nucleus and move outward throughout the cell. Look closely. Do all the tubes look the same? No, there are two distinct tubes in this network. This tube has some substances attached to it, while the other is a plain tube. These are called as the ribosomes, and the tube which has ribosomes attached to it is called as the rough endoplasmic reticulum. Yes, if the tube has ribosomes attached to it, it is referred to as the rough endoplasmic reticulum. What about the tubes which have no ribosomes attached? What's the antonym of rough? You're right, it's smooth. These tubes are called as the smooth endoplasmic reticulum. Why are they called so? Rough endoplasmic reticulum because the ribosomes attached make the surface of the tubes uneven, and smooth because the surface is even. Nevertheless, both these tubes form a network throughout the cell. What does this network form? These look like slides which will assist movement throughout the cytoplasm. That's exactly why they are placed in such a manner. The rough endoplasmic reticulum makes proteins. These proteins then have to be transported to the other parts of the cell where they are needed. Any guesses on how they will move? Yes, these proteins move through the endoplasmic reticulum to reach their destination. If the rough endoplasmic reticulum manufactures proteins, what does smooth endoplasmic reticulum do then? It helps in the manufacture of fat molecules or it synthesizes lipids. Did you know that some of the proteins and lipids they make help in building up the cell membrane? Yes, and this process has an interesting name, and it's called membrane biogenesis. Some of the proteins and lipids also work as enzymes and hormones. So the substances produced by the smooth endoplasmic reticulum helps in various ways. Do you also know that different cells of the body have smooth endoplasmic reticulum, but they all help in different ways? For example, the liver cells in animals have smooth endoplasmic reticulum. Do you know what the smooth endoplasmic reticulum does here? It helps in the detoxification process by removing the poisons and drugs present in the cells. Okay, so quickly, what all does the smooth endoplasmic reticulum do? Let us review its functions once again. The smooth endoplasmic reticulum helps in the manufacture of fat molecules. Secondly, it synthesizes lipids, and some of these molecules help in the building up of the cell membrane. The proteins and lipids also function as enzymes and hormones. And in some cells, they act as detoxifying agents of the cell. Even though the endoplasmic reticulum differs in various cells, their main function is to be a transport channel for proteins. They act as a transport network of the cell. Thus, they act as a framework for the biochemical activities of the cell. Do you think all the proteins, lipids, and other molecules formed in the endoplasmic reticulum are used only in the cell? No. The proteins produced here need to be now sent to other places where they are needed. This could be both inside and outside the cell. Let us see how that is done in our next video.
So we have understood very well about the structure and function of endoplasmic reticulum, both smooth and rough type from the video also. Right? We have discussed also before watching the video, and once again you can see that the smooth endoplasmic reticulum, it will manufacture the lipids, and rough endoplasmic reticulum that contains the ribosomes will manufacture proteins which will be utilized in membrane biogenesis. So bio means any relating to any biological organism or living organism or living molecule, and genesis means synthesis, right? So biogenesis means the production of the cell membrane. That is why the name is membrane biogenesis. Now we'll know about the Golgi apparatus. Now this Golgi apparatus we need to know was named after the scientist who discovered it, that is Camillo Golgi. So the scientist Camillo Golgi discovered it. So the name is Golgi apparatus, and this is known as the packaging organelle. As the hint given in the video of the endoplasmic reticulum, that after something is manufactured in a factory, that needs to be transported, packaged and transported. So that work is done by the Golgi apparatus in the cell. After the lipids and the proteins are synthesized in the endoplasmic reticulum, they are then processed, packaged and transported out by the Golgi apparatus. So they are known as the packaging organelle. So let's watch a video regarding Golgi apparatus and then we'll discuss further.
In the last video, we saw the proteins and other molecules manufactured by the ribosomes and the smooth endoplasmic reticulum. When products from a smaller factory have to be delivered out, do they go out without the packaging? The factory produces various goods. The spare parts are assembled, some may be modified, put together, packaged, and then sent to the market. This unit is called as the modification and packaging unit of the factory. In a similar way, the endoplasmic reticulum acts as the manufacturing body of the factory, and the Golgi apparatus is the modification and packaging unit of the factory. The apparatus is named after Camillo Golgi, who was the first to describe the Golgi apparatus. An interesting fact about him is that he found a revolutionary method of staining individual nerve cells and the structures of the cells. This method is referred to as the black reaction. In this method, he uses a weak solution of silver nitrate. This is valuable in tracing the processes and the most delicate branches of cells. One of the cell organelles is the Golgi apparatus. But how does it look? Let me show you. Have you ever seen deflated balloons? The Golgi apparatus looks like deflated balloons placed parallel to each other. What are these things here? Each of this is called a vesicle, and these vesicles placed in stacks are called as cisterns, and they are bound by a membrane. These tubes are connected to the endoplasmic reticulum. Why do you think they're connected to it? It's because the materials synthesized in the endoplasmic reticulum need to be packaged and sent to different places. Imagine a simple sugar molecule going into the Golgi body through the endoplasmic reticulum. It passes through the cisterns and gets modified. A simple sugar will get modified to be a complex sugar, and this sugar molecule is packaged and sent to the desired destination. The Golgi apparatus thus stores, modifies, and packages substances sent through the endoplasmic reticulum. They also form lysosomes. What are lysosomes? We will study those in our next video.
So let's discuss about the Golgi apparatus. So as I told you, it can be compared with the packaging unit of the chocolate factory. So after the chocolate making process is complete and it is ready to be marketed out or shipped, then the chocolates need to be packaged in different containers. So similarly, in case of cell also, as I told you before the video, that when the proteins or lipids are synthesized by the endoplasmic reticulum, then they need to be packaged. So that is the reason why Golgi apparatus remains closely associated with endoplasmic reticulum. And this has got two phases. The Golgi apparatus has got two phases. One is the cis phase, through which it receives the materials from the endoplasmic reticulum, and another on the opposite side, another phase is the trans phase, through which the materials are transported out after being packaged. So it can be the vesicles that will contain the proteins and the lipids. They will go and they will form the membrane, or they will be utilized for other processes of the cells, and also they give rise to lysosomes. Now, what are lysosomes? Lysosomes are also known as the suicidal bags. We all have heard the name. Why? Because they contain the digestive juices. So, therefore, if any pathogen or any impurity enters the cell which will harm the cell, so the lysosome will engulf it and also will destroy that particular cell as a whole, because that cell, if that cell gets damaged, then the pathogens will spread from that cell to the neighboring cells. So therefore, in order to prevent the infection from spreading, so the lysosome destroys the whole cell. So that is why they are known as suicidal bags. So we'll know about them also. So in this video, let's see about mitochondria and lysosomes. Then again we'll discuss.
We have seen that Golgi apparatus also helps in the formation of lysosomes. Do you know what lysosomes are? They too are organelles of the cell and have a specific function. What function do you think they serve? Every cell produces a lot of waste material, and waste can be harmful. Don't you think we need agents to clean the cell? Yes, the lysosomes do exactly this. They are the waste disposal agents of the cell. All the material which can be harmful for the cell is digested by the lysosomes. Yes, you heard me right. They digest the waste material. Imagine the lysosomes to be a bag that moves around the cell, carrying enzymes within its body. Whenever it comes across a foreign body or a bacteria, it will engulf it and digest it. It also digests the old and worn out cell organelles and thus gets rid of the waste material in the cell. Doesn't it sound weird? How is it possible to digest this waste? The enzymes in the lysosomes are very powerful and can digest all organic matter. Sometimes, during this process, the lysosomes do get damaged. What would they do then? A damaged lysosome is the same as an old organelle and thus is waste material. At this point, the lysosome bursts open and the enzymes digest their cell remains. Wait, so does it like kill itself? Yes, thus they are also known as suicide bags. Structurally, the lysosome is a membrane-bound sac filled with enzymes. These enzymes are made by none other than the rough endoplasmic reticulum. The lysosomes therefore form the waste disposal system of the cell. Without them, the cell will accumulate waste material and it may in turn get damaged. But where does the energy for all these activities come from? It comes from an organelle called as the mitochondria. This organelle is called the powerhouse of the cell. What does a powerhouse do? A powerhouse produces molecules that give energy. This energy is in the form of adenosine triphosphate molecules, and these are the energy currency of the cell, also known as ATP. But what does the cell use this energy for? This energy is used for making new chemical compounds and also for mechanical work. Every process in the cell requires energy, and this energy is provided by none other than the mitochondria. Do you want to see how a mitochondria looks? This is mitochondria, which is a very important organelle of the cell. It has two membranes, the outer membrane and the inner membrane. Why two membranes? The outer membrane is porous, which means it allows the transport of material. The inner membrane is interesting to understand. It has many folds and therefore has a larger surface area. What is the use of a larger surface area? A larger surface area would be a wider base for ATP generating chemical reactions. Larger the surface area, larger the amount of ATP molecules generated. However, being the powerhouse of the cell, it is a special organelle. What do I mean by special? This organelle has its own DNA and ribosomes, just like the nucleus. Therefore, they can make their own proteins. They do not depend on any other organelle for their proteins. And of course, they produce energy, which makes every other organelle dependent on them. That's all about the lysosomes and the mitochondria. We will look at the colorful organelles called plastids and vacuoles in our next video.
So you have already watched now in the video. So let's discuss about the lysosome. We have already discussed. So lysosome is also known as the janitor of the cell. Why? Because its duty is to clear the cell of the waste product. So there is no toxicity of the cell. So this is also known or comparable to the janitor of the chocolate factory that we were doing the comparison. And mitochondria is known as the powerhouse because it generates the power in the form of ATP molecules, right? And mitochondria, if we look into the structure, that you have got the idea that it has got two membranes, outer and the inner has got folds for increase the surface area for ATP generation. Those folds are known as cristae. One is crista and the plural is cristae. So these cristae are actually they bear the structures which give rise to the ATP molecules. The storehouse of ATP molecules is this mitochondria only. And we have also known from the video that the mitochondria is the structure which will contain its own DNA and ribosomes. That is why mitochondria is known to be semi-autonomous in nature. Why semi-autonomous? Because it does not run or it does not actually follow its own DNA instructions only. It abides by the instructions received from the nucleus also in the form of the proteins coded by the genes. So therefore, on one hand, it has got its own DNA to run or to function on its own and own ribosomes to synthesize its proteins. But on the other hand, it also abides by the instructions given by the nucleus. That is why it is known to be semi-autonomous in nature, and it is comparable to the power unit of the chocolate factory, which supplies, suppose for example, electricity to the whole to all the processes in the whole factory of the chocolate. Now we'll also see the another two types of cell organelles, that is the plastids and vacuoles. Now plastids are also semi-autonomous, just like the mitochondria only. They are also semi-autonomous because for the same reason, obviously that they have got their own DNA and own ribosomes to produce their own proteins, but they also abide by the instructions received from the nucleus. And vacuoles, they are also vacuoles, they are present particularly in the plant cells. They are very prominent and large. I already told in the previous lecture, they make up almost 30% of a mature cell volume of the plants, and they contain the cell sap. What do they contain? Cell sap. So this is the fluid which also has got similar functions to that of the lysosomes. That is why vacuoles are present in the plant cell and lysosomes are not so much important there, or that is why that is lysosome is not seen also. It is either almost absent or if present very less or negligible in number, because the functions of the lysosomes is actually carried out by the vacuoles only. They have got comparable functions. But since animal cells do not contain vacuoles, so therefore the number of vacuoles and their importance also is far more in case of animal cells. Now let's watch a video regarding plastids and vacuoles before going into further discussion.
There are some organelles which are only exclusive to the plant cell. Do you know what they are called? Plastids. That's what they're called. But what is peculiar about them? Plastids are colorful organelles of the plant cell. What exactly makes them colorful? These organelles have pigments inside them which give them color. However, we also have colorless plastids. You can remember them as colored and colorless, but as it's biology, they had to be given fancy names. The colored plastids are called as chromoplasts, and the colorless plastids are called as leucoplasts. There are many more subtypes which you will learn in higher grades. But in this video, we will look at these two, chromoplasts and leucoplasts. Now you should be able to answer the next question I'm going to ask you. What gives the leaves their green color? Yes, it's chlorophyll. Plastids containing the pigment chlorophyll are called chloroplasts. It's a special kind of chromoplast. This is how you should understand it. Chromoplasts that contain chlorophyll are called chloroplasts. So yes, chloroplasts containing chlorophyll, a green color pigment that gives the plants their green color. Wait, think about all the leaves you have seen so far. Have they always been green? Have you seen some plants that have yellow and orange spots on their leaves? Why is that so? Yes, the chloroplasts also contain yellow and orange pigments apart from chlorophyll. Leucoplasts, on the other hand, have no color and they store materials such as starch, oils, and protein granules. The starch produced by the process of photosynthesis is stored in the leucoplasts, and the pigment chlorophyll helps to conduct the process of photosynthesis. How does the internal structure of the plastids look? If we look at the inside of the plastids, we can see numerous membrane layers placed in some material. What is this material called? This material is called the stroma. It is like the cytoplasm in the cell in which the plastids are embedded. Plastids are similar in their external structure to another organelle of the cell. Can you guess which that organelle is? Yes, the mitochondria. The mitochondria and the plastids have similar external structures. Plastids also possess DNA and ribosomes like the mitochondria. Another important organelle present in both the plant and animal cell is the vacuole. What are vacuoles for in a cell? Vacuoles act as storage centers for the cell. They are storage sacs for the solid and liquid components of the cell. However, their size differs in the plant and the animal cell. Do you recollect the difference between the two? vacuole which may occupy more than half of the cell volume. Okay, it's a storage unit. Great. What are the substances that the vacuole stores within it? The vacuole stores amino acids, sugars, proteins, and various other substances within itself. Also, it must be noted that in some unicellular organisms, this vacuole is instrumental in expulsion of excess water and waste material from the cell. Thus, a vacuole may serve many different functions.
So therefore, we have known about plastids and vacuoles. Now discuss a bit about them and summarize. So plastids are of three types. We have just now seen, that is the chloroplast containing the chlorophyll, which absorb the solar energy during the photosynthesis, and then there is chromoplast, which imparts the other colors of the plant, such as the red, yellow, brown, orange, all these colors, and leucoplasts, which are present in the parts which are actually, they do not impart any color, they act as the storage organs for starch, proteins, and fat molecules. So these are the types of plastids, and I have already mentioned they are semi-autonomous, just like mitochondria. They are also double membraned in their structure, and the plastids, within the plastid, if you look into the structure, the disc-like structures which are stacked one above the other, they are known as grana, they are plural, or granum, singular, right? So these are the sites where a part of the photosynthesis called the light reaction that takes place. Okay, and then the stroma is another part or of the plastids, the part of the internal structure which acts as the cytoplasm of the cell, in which another part of the photosynthesis known as the dark reaction takes place. So that is the stroma, which is which is comparable to the cytoplasm, right? And the stacks of grana, they are attached, there are not only one stack, there are many stacks such as this, which are attached to one another by some connections. They are known as intergranular lamellae. Again, if it is singular, it is lamella, and if it is plural, it is lamellae. So therefore, the connections, they actually connect the stacks with one another, so that the whole process of photosynthesis that takes place within the chloroplast, that becomes a synchronized process. So this is the structure of the plastids and also the functions of the plastids. Now these plastids are particularly for the plant cell, we know, and we have discussed when we had actually seen the differences between the plant and the animal cell. For both the plastids and the vacuoles, they are important parts of the plant cell only. So since plastids are the sites of photosynthesis, particularly the chloroplast, so that is actually compared to the kitchen of the chocolate factory. And vacuole is the warehouse because it stores many things, as I told you, in the cell sap, and also the boundary or the border of the vacuole is known as tonoplast. Inside it has got cell sap, I already told, and the boundary is known as tonoplast, which contains the nutrients as well as some of the waste substances also. It is filled with watery fluid containing various substances, and one more function is this, that to create turgor pressure, which gives more support and rigidity to the plant cell. Now, what is this turgor pressure? We know when water enters the cell, then the entry of the water through the semi-permeable membrane of the cell creates a pressure. Water enters the cell through the process of osmosis. That is why the pressure created by the entry of water molecules through osmosis is known as osmotic pressure. Now, this osmotic pressure can lead to collapse of the cellular integrity or the cellular structure. That is why, to counteract that pressure from outside, so a pressure should be given as the opposite reaction to the osmotic pressure in order to maintain the rigidity and support of the cell and the whole structural integrity. So therefore, since the vacuole occupies almost half of the space of the plant cell and contains the cell sap, so it provides a opposite directional pressure, that is the turgor pressure. That is why it provides turgidity to the structure of the plant cell. Turgor pressure provides turgidity. So this pressure helps to maintain the cellular structure against the osmotic pressure, since it is in opposite direction. So these are the important functions of vacuole. We already have dealt with these differences in our earlier class. So how lysosomes clear out the worn out cells? You can say if the foreign cell enters or the damaged organelle is there, so lysosome will digest that, and then it will carry that outside the cell, and all it will digest it with the help of the digestive enzymes present inside it. Now, human body is composed of more than 30 trillion cells, but all the cells are not alike. So, how are so many cells formed in our body? Obviously, we have got the process of cell division, through which new cells arise from the pre-existing cells. Now, in case of unicellular organisms, you can see here that there is simple binary fission, just like amoeba, that in which the organism divides into two daughter cells, that is two daughter cells. So this is a process called binary fission. Binary means two and fission means division, right? So that is a simple type of, and this is cell division and also reproduction, because in case of unicellular organisms there is only one cell. So if the one cell divides and form two daughter cells, then obviously the cell number as well as the organism number both are increasing simultaneously. So that is why it is a process of cell division as well it is a process of reproduction. But it is not so in case of multicellular organism, because we are made up of many cells. So even if some of the cells divide in our body, it's not that the whole organism is being born from one organism. And moreover, we have got the sexual mode of reproduction. This binary fission is the asexual mode of reproduction, because here there is no production and fusion of male and female gametes. But here in case of multicellular organism, we have got production and fusion of male and female gametes, then fertilization, formation of zygote, and then the new embryo of the new offspring is then given birth. So let's summarize. Cell wall is the outer covering of the cell that protects the cell against the mechanical injuries, then the wear and tear, and also the invasive pathogens. Then comes the cell membrane, which is permeable in nature, semi-permeable, unlike the cell wall, which is freely permeable, and it regulates the entry of some of the molecules, and it also acts as the outer covering which encloses the cell contents. The cytoplasm is the main area or the floor for it acts as the medium for the cellular activities. Mitochondria supplies the energy in the form of the energy currency, that is the ATP. That is why it is known as the powerhouse of the cell. And vacuole acts as the storage or the warehouse of the cell, which stores the nutrients, and also it helps to maintain the turgidity and structural integrity by providing the target pressure as an opposite reaction to that of the osmotic pressure. In nucleus, it is the brain of the cell. It controls the functioning of the cell and all the metabolic activities. ER is known as the skeletal system of the cell. Yes, ER is the skeleton, since it forms the skeletal framework of the cell. So it is known as the skeletal system of the cell, and we know they are involved in the synthesis of proteins, if it is rough endoplasmic reticulum, and of lipids, the smooth endoplasmic reticulum. Ribosome is the actually the protein factory of the cell, and Golgi apparatus is the packaging unit of the cell, and it also helps to give rise to lysosomes, which are suicidal in nature and removes the unwanted substances and helps to maintain the toxin free nature of the cell. Plastids, they actually synthesize different types of the colored pigments and also helps in photosynthesis, and also leucoplast actually acts as the storage house for the lipids, starch and proteins. Then centrosome, this is also another organelle present in the cells, that it is helping the cell to divide. When the cell division occurs and there is duplication of the genetic material before there is division of the nucleus, so what will happen, the centrosomes actually pull apart the two sets or two copies of the genetic material from one another, and then only the nuclei and subsequently the cytoplasm and the cell divides to form two cells from one cell. So therefore, centrosome is a very vital organelle for cell division and cell, we can say, increase in number. And this centrosome is absent in one type of cell, that is the neuron. That is why nerve cells cannot regenerate. So this is the comparison table of chloroplast, chromoplast and leucoplast. You can refer to this for their pigment that they contain and also their functions. So we have already discussed our eukaryotic and prokaryotic cells in our last lecture. So this is the brief summary in the form of comparison. Now coming to the last thing, that is the cell division. So what is cell division? So cell division is a process in which the cell will divide and form new cells. So therefore a typical cell which contains, in case of humans, we have 23 pairs of chromosomes. That means 23 into 2, 46 number of chromosomes. Now when a cell contains all the 46 number of chromosomes, that is known as the diploid stage of the cell. That means we have inherited 23 number of chromosomes from father and 23 number of chromosomes from mother during the process of fertilization. So that then when they combine in the zygote, they form the 46, that is the diploid state, right? So all the body cells, we have 22 pairs of the somatic cells, that 22 pairs of somatic cells. That means 22 pairs of chromosomes in the or the somatic chromosomes, not somatic cells. These are the somatic chromosomes. What will happen there, that these 22 pairs of somatic chromosomes or 44 number of chromosomes will code for other types of proteins which will dictate how will be our the body color. For example, the height, the weight, the actually the body structure, everything. And the remaining one pair, that means two chromosomes, they are known as sex chromosome. Now, depending on the type of sex chromosomes we have, now our sex is decided whether will be male or female. So therefore, we know that the female body has got ovum as the female gamete. Now, ovum always contains only X type of chromosome, right? And the male gamete, which is present in the male reproductive system, that is the sperm, contains either X or Y type of chromosome. Now, if the ovum containing X type of chromosome combines with sperm containing X type of chromosome, then we get the combination of XX, and then the child born will be girl. On the other hand, when the ovum containing the X chromosome combines with the sperm containing the Y chromosome, then the combination is XY, and the child born will be a boy or a male child. So therefore, from this combination or the clear description, we can understand that the father is responsible for determining the sex of the unborn child and not the mother. So this was the notion which has been cleared out by the scientists through their actually experiments for all these years, because earlier many social malpractices were there regarding this issue, but nowadays it is very clear that the father is the one responsible according to the sperm that he is donating or that is combining with the egg cell of the mother. So that is the thing. And now the sex cells, when they contain, I already told you that the gametes, the ovum and the sperm, the ovum, which is the female gamete, and the sperm, which is the male gamete, right? So they combine with each other. Now I already told you each of them will contain 23 number of chromosomes, and when they combine they will give rise to 46 number of chromosomes. So now 23 number of chromosomes when present in the female gamete or the male gamete, this is known as the haploid stage. Haploid stage, this end stage, right? That is the only one set. That means one set from the father and one set from the mother. One set means consisting of 23 number of chromosomes, and diploid stage consisting of 46 number of chromosomes. See? So therefore, now cell division is of two types. One is mitosis, another is meiosis. Let's understand that. So mitosis is when the cell will develop the chromosomal material. They will make clones. For example, if there are 46 number of chromosomes, so before going into mitosis, the cell will make up another set of 46 number of chromosomes, and then the nucleus will divide, and then the cell membrane will divide. So then that is known as the mitosis. That is simple division in which there will be duplication of the genetic material and then cellular division. That is plain increasing number. So this is one type of cellular division. And what happens in case of meiosis? In case of meiosis, from the diploid stage, that is the 2N, it gives rise to cells, two cells only. Here also we had got two cells, but each cell had the diploid state only. On the other hand, in meiosis, each cell will give rise to two cells, but they will have the haploid, that means N. So therefore, if we look at the diagram, from one cell, two daughter cells will be formed, which will be of diploid state only in case of mitosis. But in case of meiosis, from the diploid cell, first it will get converted into another type of cell which can undergo into the nuclear division and give rise to haploid number of cells. So, and then they will divide to form two daughter cells each. So, therefore, from one cell, one diploid cell, we can get 1, 2, 3, 4 number of haploid cells. So, this is the case of meiosis, and this happens in the when the cells form the gametes, such as the male gamete and the female gamete. That means the sperm and the ovum respectively. So here chromosome number remains the same, but here the chromosome number is halved. Means if it was 46 before, it will be 23. Now it happens after two nuclear divisions. That is why four daughter cells are formed from one diploid cell. But it happens in case of mitosis after one nuclear division only. That is why only two daughter cells are formed from one diploid cell. So that is all from this chapter. I hope I could clear out the topics that have been delineated here. But after this also, if you have got any queries or doubts, do let me know and post them in the comments below. And we'll meet again with another topic of different chapter in the next class. Till then, stay tuned. Take care. Bye-bye. Thank you.